Hello there, and welcome to this Biology Mind A-Level tutorial on cell division, where we will be having a look at the phases of mitosis. So just to recap what we did in the last lesson, we learned about the cell cycle and how it is a regulated sequence of events that occur during cell division. We looked at how there are three phases of the cell cycle: number 1, interphase; number 2, mitosis; number 3, cytokinesis. And you now understand that the end product of the cell cycle is the division of one parent cell to form two genetically identical daughter cells. And we also understand why cell division is essential for ensuring the survival of an organism. So today in this tutorial, we have two key learning objectives. Number 1, the phases of mitosis, and number 2, an experiment to investigate mitosis. So there are 5 phases of mitosis. Number 1, interphase; number 2, prophase; number 3, metaphase; number 4, anaphase; and number 5, telophase and cytokinesis. So let's have a think about where we are before we enter mitosis. So we have just undergone interphase, where we have doubled the DNA, as you can see here, and we have also doubled the organelles in the parent cell. So we're quite ready for mitosis to occur and to split that DNA and organelles and proteins between the daughter nuclei. So after interphase, before interphase, our parent cell is diploid, remember, because human cells are diploid. They have 2 copies of each chromosome. From chromosome number 1 to chromosome number 23, they have 2 copies of each. So we refer to that as 2N. It's a diploid cell. Now, after interphase, we now have duplicated our DNA. So we've now got 2N multiplied by 2. We know that humans have 46 chromosomes, or 23 pairs. Now, 46 chromosomes times 2, because remember we replicate our DNA in interphase, equals 92 chromosomes. So after interphase, the cell is tetraploid. It's got 92 chromosomes, or 4N. So we met this term a couple of tutorials ago when we were talking about DNA and chromosomes. Now we've duplicated our DNA, but overall these chromosomes will be evenly split in half again during mitosis, with each daughter cell again now having 2N chromosomes, so having the diploid number of chromosomes, which is 46, or 23 pairs. Okay, so you can see that mitosis results in daughter cells that are genetically identical to the parent cell and have the same amount of genetic information as the parent cell. So during that S phase of interphase, the cell replicates DNA, which results in 2 copies of each chromosome that you can see here. So this cell is now tetraploid, whereas this one was diploid before those DNA or chromosomes were copied. And we have our homologous pairs of chromosomes. Now they have formed 2 pairs of homologous chromosomes. And then these chromosomes are split in mitosis, and each daughter cell gets 46 chromosomes. So let's have a look at the stages of mitosis. So number 1 is prophase, which is where the chromosomes condense, which makes them shorter and thicker, making them visible as 2 sister chromatids. So remember, this is one sister chromatid, and this is the second sister chromatid. One sister chromatid, the second sister chromatid. And this white dot in the centre is our centromere. Then what happens is centrioles, which are these things, split and move to opposite ends of the cell, and they produce a mitotic spindle, or spindle fibres, which are these things branching out from the centrioles. So these are our spindle fibres, and this is our centriole. Meanwhile, the nuclear envelope disintegrates and the chromosomes are freed into the cytoplasm. So those are our stages of prophase. First of all, our chromosomes condense, our centrioles move to opposite poles of the cell, so opposite sides of the cell, and they produce a mitotic spindle, so these spindle fibres, which extend across the cell towards the nuclear envelope, but that nuclear envelope is meanwhile disintegrating, and those chromosomes are being freed into the general cytoplasm. So that was prophase. Now we come on to stage number 2, metaphase. So by this stage, the nuclear envelope has disintegrated, that it has gone, and the spindle fibres attach themselves to the centromeres of each chromosome. So you can see the spindle fibres have extended further towards the centre of the cell, and they've attached to the centromeres of the chromosomes. And the chromosomes have lined up in the middle of the cell, which we actually refer to as the equator of the cell. And then what happens is those spindle fibres contract and pull the chromosomes, lining them up at the metaphase plate. So you can see those spindle fibres contracting in anaphase, and they pull the sister chromatids away from each other to opposite poles of the cell. So sister chromatids are pulled apart to opposite poles or opposite ends of the cell. And this splits the chromosome into 2 V-shaped sister chromatid structures. So let's have a look at what comes after that. So then it's telophase. So the spindle fibres have pulled the chromatids to opposite poles of the cell, as you can see here. They're on opposite poles of the cell. The chromatids uncoil and can no longer be called chromosomes, and the nuclear envelope reforms around each set of chromosomes. So the cell now has 2 nuclei, you can see them here, each with a full set of identical DNA, and meanwhile the cell prepares to split the cell membrane, cytoplasm and its contents via cytokinesis. And that's how we get our formation of our 2 daughter cells. Once this splits in the middle here, this is one daughter cell and this is our second daughter cell. So cytokinesis is that final step of mitosis, and we've now got our 2 daughter cells, and each daughter cell is identical to each other and their parent cell. And note that each daughter cell has 1 nucleus, a complete set of DNA and a set of organelles, as well as the same amount of cytoplasm as their sister cell. So a really good tip here is that it's really important you understand the duplication and splitting of chromosomes in each stage, and really note how that daughter cell ends up with the exact same genetic material as the original parent cell, as well as to its other sister cells. And that is the exact same genetic material as the original parent cell before interphase occurred. So this will sum up exactly what we've talked about. So it might be a really nice idea to jot this down in your notes and draw this diagram and then label it up with everything that we talked about in each of these stages so that you really remember what has happened here. Okay, and then again, we have a nice summary of our cell cycle. So number 1, the cell grows and makes a new set of organelles and proteins for the daughter cells in that G1 phase of interphase. Remember this bit here, oops, excuse my wobbly circle drawing, but that bit here is interphase. Okay, which remember is phase 1 of the cell cycle, but it's split into G1, S and G2. So in G1, we've got that new set of organelles being made in proteins for the daughter cells. In stage S, stage 2, we have cellular DNA being replicated. And then in stage 3, cell growth continues and there is synthesis of special proteins in preparation for mitosis. Then over here we have mitosis happening, where the cell splits its DNA and organelles into two identical daughter cells, and cytokinesis is that actual splitting of the cell into its two daughter cells. Okay, so we can investigate mitosis using a practical, which focuses on using something called root tip squashers and an optical light microscope. So we're going to go over the stages of this practical. So number one, we warm hydrochloric acid in a water bath at 60 degrees Celsius. Then we cut 1 to 2 centimeters from a growing root to get the root tip, which is the active region where mitosis is occurring. So this is what we're interested in. So we can cut the root using a scalpel. And the growing root tip can be from any plant, such as a potato or onion. Then we wash the root tips in cold water and dry them on filter paper. And we transfer the root tip to the warm acid for about 5 minutes. And be careful when you do this, because obviously we are using acid here, which can hurt you. Okay, and then we remove the root tips from acid and wash them again in cold water and dry on filter paper. And then we place the root tip onto a clean microscope slide. And we cut around 2 millimeters from the end of the growing root tip. And you can discard the rest, but keep those tips. We then stain the root tip and leave for about 2 minutes, and the stain binds to chromosomes, enabling them to be visible under a microscope. We then use a mounted needle to break up the tissue and spread the cells of the root tip out onto the slide. Then we cover that with a cover slip, and we press down on that cover slip firmly, because the further we press that root tip, the thinner we make it, and the thinner we make it, the more easily light passes through our sample, which will make it easier to have a look at the cells under the optical microscope. And we then use that light microscope to visualize chromosomes in the tips, and you should be able to see different cells in different stages of mitosis. So for example, over here, let's say this is what we can see under our light microscope. Over here, you can see non-dividing cells. Now, we know that because their chromosomes aren't really that visible, and their nuclei are nice and whole, and there's a nice clear nuclear envelope. Then over here in B, we can see nuclei preparing for division. So we know this because we can see that our chromosomes have condensed now, and they've formed nice thick pieces that we can see. And we can see that they are starting to line up at various parts of the cell. Then over here, we can see mitotic figures. So we can see metaphase and anaphase occurring, where those sister chromatids are pulled apart. And then over here, we can see a pair of daughter cells shortly after division. So this is how you can have a look at mitosis under a light microscope. You can use something such as a root tip where mitosis is happening very much so, and we can look at that under the light microscope and have a look at the different stages of mitosis going on. And look, you can see there's a difference, even a further different stage here, where in anaphase, where those chromosomes are being pulled apart to opposite poles of the cell. So it's really, really interesting. I hope you get the opportunity to do this in your lessons. Okay, so in the exam, they are unlikely to ask only factual questions. They want you to understand the method and not to just regurgitate it. So we've come up with some questions to have a go at. So we'll talk through them, but if you want to give the video a pause at this point and have a go yourself, feel free to. But we'll talk through it now if you just want to sit back and listen. So why do we use multiple root tips? Well, this is quite a simple answer. So the root tips are very delicate and can decompose, so using several increases the chances of getting good images at the end. Number 2, why is acid used? So this is because plant cells tend to stick together due to their cellulose cell walls, and the acid separates the cells, enabling the stain to enter more easily. And actually warming the acid speeds up this process of that stain entering the cells. Now, what does the stain bind to? So different stains can be used, but many bind to chromatin DNA. And then finally, how would you change the method to improve your end images? So you could do this in multiple ways. Just have a little brainstorm. So if I were to brainstorm, it would be increase the time spent putting those root tips into the hydrochloric acid, increase the temperature of the incubation, again to speed up that process, and then increase time spent in the stain because that's going to increase the visibility of our chromosomes in our sample. Okay, so well done following through with that tutorial. We've covered quite a lot there. Hopefully you get the chance to do that experiment with the root tip cells in your lessons, because then that will give you a really good opportunity to have a look at mitosis as it is happening, which is really exciting. Okay, I will see you for the next tutorial.